Our next speaker is Dr. Yoshi Nishikawa, who comes to us together with Dr. Harari, traveled far to get to us, so thank you. Special thanks for that. Dr. Nishikawa received his degrees in an MD and a PhD from the University of Mia. I'm only trying this once. And he did his part of his training with Dr. Lloyd Old at Memorial Sloan Kettering in New York, and Dr. Shimon Sakaguchi at Osaka University. And currently, Dr. Nishikawa has the title of Director of the Division of Cancer Immunology at the Research Institute and Exploratory Oncology Research and Clinical Trial Center in Tokyo, Japan. Thank you so much for joining us.
Well, first of all, I'd like to thank BD for inviting me this exciting meeting. So this is our institute, National Cancer Center. So National Cancer Center has two hospitals. So this is Central Hospital and East Hospital. So actually, our Central Hospital is just in front of Fish Market, Tsukiji, but unfortunately, Tsukiji will move soon. So due to the Olympic Games, so we are now very sad for their moving. But anyway, so back to my talk. So first part, I'd like to just touch about why the importance for analyzing the tumor microenvironment by flow cytometry. Second part is our collaboration with BD Japan for developing tumor-infiltrating lymphocyte. Well, the last part is how the importance to combine the immune assays of the genetic assays and flow cytometry assays. The first part I'd like to mention about why the flow cytometry assay is critical for analyzing the immune responses in tumor microenvironment. So before going to that, so just this is the history of cancer immunotherapy. So about 50 years ago, so by Dr. Barnett, so hypothesis of cancer immunosurveillance was provided. After about 50 years later, we now know their expansion to the cancer immunoediting hypothesis by Dr. Bob Schreiber and also my mentor, Dr. Lloyd Old. And also blue one is a history of a cancer immunotherapy, starting with probably with a choline and IL-2 and other cytokine therapy, now the immune checkpoint inhibitors. So now the cancer development is summarized as the 3 steps by Bob Schreiber and Dr. Lloyd Old and Mark Smith. So as I call the cancer immunoediting. So there are 3 steps of the cancer development. First, elimination, equilibrium, and escape phase. So elimination phases are all previously known as cancer immunosurveillance. During our life, we experience several stimulations like UV and other viral infections and inflammation. Then abnormal cells are generated, but of course the cell is themselves renew or have their own treatment strategy to come back to the normal cells. But unfortunately, still cells cannot go back to the normal. The immune system eliminate these abnormal cells from the host. But meanwhile, abnormal cells gain their several methods, several strategies to survive in the host, even in the immune competent host. So by reducing the antigenicity, so-called equilibrium phase, and also recruiting our immune suppressive cells, immune suppressive components for developing as the clinical cancers. So among them, we focus on the several immune suppressive cells and molecules, particularly immune suppressive cell regulatory T cells. So you know that regulatory T cells are known as was found by also my mentor, Dr. Shimon Sakaguchi, as a CD4 positive, CD25 positive cells in 1995. But at that time, Tregs are considered to suppress directly to helper cells and killer cells. But now we know that the regulatory T cell mainly suppress the antigen-presenting cells by reducing their antigen-presenting activity. So now also we know the more comprehensive regulatory T cell function for suppressing, inhibiting the antigen-presenting capacity, and also consuming interleukin-2 for T cell survival, and also controlling T cell metabolisms. So among them, so using these comprehensive various strategies, Treg cells control the immune suppression, control the activation of effector T cells. In cancer setting, we know the FOXP3 positives are considered to be associated with a worse prognosis and various types of cancers, for example, melanoma and other types of cancers. Colorectal cancers, we know the FOXP3 positive cells are associated better prognosis in several various previous reports. But we saw that this is very strange. Why in colorectal cancer FOXP3 positive cells show the opposite role compared to other types of cancers? Then we analyze the details of the FOXP3 positive cells with flow cytometry assays. So long story makes short, now we can analyze a regulatory T cell like this way. So a previous speaker already introduced this analysis. So using the CD45 RA and FOXP3, we can divide the FOXP3 positive cells in 3 fractions, fraction 1, 2, 3. Among them, this fraction just came from the thymus, and then after activation, they move to here, FOXP3 high, CD45 negative fraction. But the important point is in humans, not in mice. FOXP3 low, CD45 area negative fraction, these fractions are not regulatory T cells. Rather, they produce cytokines. They are like Th1 and Th17 cells. In cancer setting, peripheral blood, slightly fraction 2 are the number of frequency of fraction 2 cells are increased in tumor tissues, as already see in the first speaker's data. So this fraction is fraction was very increased in tumor local site. But this is generally observed in melanoma and renal cell carcinoma. How about in colorectal cancers? We analyze using our colorectal cancer cohort. Then we now know that there are 2 types of colorectal cancer from immunological phenotypes. One is similar to melanoma, like most of the FOXP3 positive cells in tumor tissues belongs to here. So, but in more than half of colorectal cancers, so tumor-infiltrating lymphocytes are stained like this. More than half of FOXP3 positive cell belongs to here, so non-Treg fraction. In that case, if you analyze with these cell with immune histochemistry, that is problem. Even in these 2 types, FOXP3 positive cell stains as well, is almost similarly stained. So we cannot distinguish these 2 types by the immune histochemistry. We need a flow cytometry assay to distinguish these 2 fractions. So then also we analyze the function of this FOXP3 low and FOXP3 high fractions. As already we have shown in the peripheral blood data, even in tumor tissues as well, this fraction, blue one, they do not have immune suppressive functions as well as in peripheral blood. So they are not Treg as well in tumor tissues, in colorectal tumor tissues. Then we try to find molecules which is associated with these 2 types of colorectal cancers. Now I call the one types which do not have these fractions 3 non-Treg fractions with FOXP3 expression. I call this type A. So another fraction, they contain FOXP3 low inflammatory cells in tumor tissue. I call the fraction 2. Type B colorectal cancers. In that case, type B colorectal cancer, they have very high expression of inflammatory signal and immune response signals. So we further analyzed in detailed molecules and found that TGF-beta, TNF, and IL-12 are critical factors for dividing these 2 types. Particularly double low fractions belongs to type A colorectal cancers. But double high fractions belongs to type B fractions. So before going to the FOXP3 role in these two fractions, just I mentioned about the prognosis of these two type of cancers, particularly IL-12 high type B, type B fraction has a very better, favorable prognosis compared to type A fractions. So then we looked at FOXP3 positive cells, role of FOXP3 positive cells in our cohort. When we looked at just the FOXP3 positive cells in entire fraction, FOXP3 is not a prognostic factor in our cohort. But when we looked at this here, so in this case, FOXP3 is associated, almost derived from a regulatory, the bona fide regulatory T cells. In that case, FOXP3 is clearly associated with an unfavorable prognosis of colorectal cancer patients. But when we looked at this type, more than half of FOXP3 positive cells are not regulatory T cells. In this case, FOXP3 high is associated better prognosis because more than half of FOXP3 positives are T helper cells. So we have to be careful about just analyzing one molecule with genetical assays of provide a very different answer for the prognosis in cancer patient. So just I quickly attach the mechanism why these two types of cancer developed. So we then analyze the cytokines, how cytokines are influenced on this FOXP3 low fraction. Then found a combination IL-12, the TGF-beta highly produce this fraction from a naive T cells, naive helper T cell fractions. Then why such cytokines are provided in colorectal cancers? Then we analyze the microbiome in colons. Then found type B fraction. They have a high attachment over microbiome, particularly Fusobacterium was highly attached to this type B colorectal cancers compared to type A colorectal cancers. So so as we know, the Fusobacterium is associated with the inflammation and also tumor genesis using these two signal E-cadherin beta-catenin signaling. And then we analyze type A and type B signaling. So cyclin D1, NF-kappa B, inflammatory signaling, tumor genesis signaling are highly detected in type B colorectal cancers. So based on this, we now we consider that colorectal cancers are immunologically divided two types. One is developed as well as other types of cancers, but another is a colon-specific cancer development. So inflammatory Fusobacterium induce our cytokine production. This cytokine influence the T cell activation, and FOXP3 low fractions are detected in these types of cancers. So this is the why that we need to analyze the tumor TILs by flow cytometry. Otherwise, we may induce a very wrong answer to the T cell infiltration in tumor tissues. The second part is in collaboration with BD Japan for the development of a biomarker in PD-1 blockade therapies. So I don't mention too much about the importance of biomarkers because cancer immunotherapy provide a long durable clinical responses, but only 20% of patients have such benefit. So we need to stratify these 20% of patients before treatment. So then we analyze, still the PD-L1 gene mutations are not sufficient for biomarkers. So we go back to the immune responses. Also, this was talked by the previous speakers. I don't mention too much, but we now know the two different types of antigens. One is neoantigens derived from gene mutation. The other is shared antigen derived from almost self-antigens. So we focus on the differences of our antigen-specific T cell activations and also suppression by regulatory T cells. So you know the self-antigen generally have a lower TCR affinity. So foreign antigens, such as viral antigens and also neoantigen-specific T cells, has a higher TCR affinity. So based on this, so we analyze their immune suppression by regulatory T cells. So Treg cell, first we analyze the specific T cell activation focusing on self-antigen, self-shared tumor antigen-specific T cells. So we labeled the CD8 T cells before activation, then added a titrated dose of regulatory T cells and try to see the activation status of these melanate specific shared self T cell antigen specific CD8 T cells. So this is the, I'm sorry, something here. So this is the specific T cell induction detected by melanate tetramers. So by adding the regulatory T cells, specific T cell induction was inhibited. But the important point is here, we labeled the CD8 T cell before cultures. So we can know how many times these cells are divided during the activation. So as you see here, without Tregs, these melanate-specific CD8 T cell can divide it more than 8 times. But the important point is here, when we added the regulatory T cells, so these T cells divided once and then stopped further proliferation. So I don't have show the detailed data, but they are in very strong anergic state. We cannot convert with high dose IL-2 and blockade over PD-1 and CTLA-4 antibodies. So these self-shared antigens are highly suppressed, strongly suppressed by regulatory T cells, cannot convert it again to the activated T cells in healthy individuals and also cancer patients. So this is the phenotypes of these anergic T cells. So they have high expression of CTLA-4 and still maintain the CCL7 and CD45 allele expression. They are in anergic state, but having their expression of CTLA-4, very strange immunological phenotypes. But how about the neoantigen-specific T cells? So this is the final goal of our collaboration, clinical application TLS assay BD&I for 20-something. 2020 is the year of Olympic Games in Japan. So still we have just made a first step, so we need to know many things before going further assays, but particularly the last part, analyzing tumor microenvironment with flow cytometry is a very critical component for biomarker assays and also cancer immunology experiment. So this is the collaborators and in National Cancer Center and also my cross-appointed university in Nagoya University. Thank you very much for your attention. I'm happy to take your questions.